Now we have Grace and King online. Seven. Any guesses what that is? It's a thing that both prokaryotic cells and eukaryotic cells controls what comes in and out of the cell, therefore maintains homeostasis. This is too early. If you don't overthink it, it's actually pretty easy. It's a cell membrane. So all cells have a cell membrane, which obviously controls what comes in and out. Of course, you could get trickier if you were overthinking it. You might think, all right, so what is it? A cell membrane that's in control of what comes in and out. But there's parts of the cell that help the membrane maintain its shape. And I'll bring those up briefly, hopefully in today's lecture, but I will not be testing that deeply. So again, like all the important parts of the cell, as far as the semester is concerned, they're all important, but as far as this semester is concerned, all the important parts we're going to learn about later after this chapter 2. So. Morrison is oh, there you go. So the cell wall, I don't know if they just say it and I missed it, but not all cells have cell walls. Like animal cells don't have cell walls. And because this is 100 level class, we generally only compare and contrast animal cells versus plant cells. Plant cells do have a cell wall, and that obviously would help maintain the structure. But animal cells do not have a cell wall. See, this is another great example. This chapter, I'm going to briefly discuss these, and they are a little bit more important because we're going to have a whole chapter about cellular division. And when we talk about that, that's when these parts are going to be more important, in which case we'll talk more about them at that point. So again, this chapter that we're covering now is just scratching the surface, a nice preview of the important stuff we're going to learn earlier or later. And that way, when you do learn them later, it won't necessarily be a review, but it won't be the first time you've seen these things. Once again, I'm going to briefly talk about chloroplasts in this chapter. But once we talk about photosynthesis, we're talking a lot more about chloroplasts. that we talked about on Wednesday. So we've already talked about cytoplasm. And that's a great way to describe it. It's everything inside the cell other than the nucleus. Well, actually, I don't really like that description either. That is kind of vague because there's stuff in the cytoplasm. Yeah, it's a semantics conversation. I won't muddy the waters. We'll briefly talk about those today. Um, Later in the semester, when we talk about how proteins are made, we actually bring it up again, but we don't get too much into the actual structure and function of the ER. It's more like we just bring it up, like, by the way, this is all happening in the ER. We'll bring this up quickly. Not very important in the grand scheme of the semester. It's like we have a small intimate gathering today. It's been a rough time at the university. This is the last time I see you guys. I bet that had something to do with it. Well, it's 8 o'clock. Let's get started. I haven't seen that video before. I just like to go through them to see which ones are worth sharing. Attendance very quickly. Of course, everyone here is accounted for, but since we're recording, this is more of me saying this out loud, so it's being recorded. Key is not here. Blount is not here. Boggs is here. Bonner is online. Clay is not here. Clayton is not here. Dawson is not here. Ellis is not here. Paula is here in red. Green will not be here today. Harrison is here in red. Henson is here in red. Last name James is not here. Kesterson is here. King is online. Lewis is not here. Lindeque is not here. Be gone for sports. Morrison is here. First name Grace is online. Last I saw. Rice is dropped. Ward will not be here today. Wilcox is not here. Wilder is not here. Parks is not here, but I bet she will be soon. Did I miss anybody? Okay. So the first word for attendance, like I mentioned, it's been a rough couple of days for the university. So in honor of people we lost. The first word for attendance is Harrington. Last name Harrington. A student athlete who was just killed. Here we go. Sorry to bring up something sad, and then all right, let's pretend like that didn't happen and get into some fun biology. But the good news is, this part of biology is not that fun. I'm just kidding. Everybody likes different parts of biology. This, for me, is not that fun. I like other stuff. But without this stuff, the fun stuff that I enjoy wouldn't be possible. But anyway, I think we left off talking about cytoplasms. And as if you were here earlier, you would have just heard that video. I guess one way you could describe a cytoplasm is it's everything other than the nucleus, right? Everything other than the nucleus. Or, as your textbook describes it, it's the contents between the membrane and the nuclear envelope, right? It's the same thing I just said, but in a different way. It's made up of the organelles that are suspended in the gel-like cytosol. And as you, again, from that video, you heard cytosol is basically like that jelly-like liquid where everything is suspended in. It is about 70 to 80% water, but it's mostly a semi-solid consistency, cytosol that is. And many metabolic reactions take place here. So for the exam, let's see, you should know what the cytoplasm is in general. Like, let's see, you know that it's basically the contents between the membrane and the nuclear envelope, aka everything other than the nucleus. You should know that it's mostly consisted of the cytosol, which is you don't need to know that number, so I'll put it next to that. But you need to know it's mostly just liquid, right? Semi-solid consistency. So cytoplasm, everything but the nucleus. It's got this jelly-like gunk in it called cytosol. Any questions about that? And again, I'm going very light on the exam with this stuff, because the stuff that's important for the grand scheme of this semester will be covered in later semesters anyway. Cytoskeleton, another great example of that. I'm not sure I'll have any questions about this on the exam, definitely on the quiz. But anyway, the cytoskeleton is a network of protein fibers that helps maintain the shape of the cell. Not only does it maintain the, like, imagine, have any of you ever seen the inside of the Statue of Liberty? You know, so it's got a bunch of scaffolding and things keeping it up, but the same kind of thing, right? Just things inside that are just kind of helping it maintain the shape. Not only does it do that, but it also secures certain organelles in specific positions. So instead of those organelles just floating around in the cytosol, right? Sometimes some of them are secured thanks to the cytoskeleton. It also allows the cytoplasm and vesicles to move within the cell. So again, if things need to be moved around, they can't move around willy-nilly. They need to attach to something that moves them, and that would be the cytoskeleton. And it enables unicellular organisms to move independently. So when you think about things like amoebas or flagella, they either have like one really long whip, which would be the flagella, or a bunch of tiny little hairs like a cilia. That's what helps them move. But again, there won't be many questions about this for the exam. If anything, just know the cytoskeleton is the thing that helps maintain the shape of the cell. And yeah, it does all those other things, but I think that's the most important. Because by doing that, by helping maintain the shape of the cell, all these other things are possible. Anyway, I guess you could say that the other way around too. It's all a matter of perspective. By allowing all these things to move around all over the cell, that also maintains the shape of the cell. But that's what I'm focused on. Any questions about this? All right, there's 3 types of cytoskeleton. I'm really not sure I'm going to ask you about this, but we'll discuss them briefly anyway. So I'm going to introduce them to you now, and then we'll look into more detail in the future slides. But the first type is called a microfilament, also known as actin filaments. Then we have intermediate filaments, and then we have microtubules. So let's talk about all 3 before I ask you if you have any questions. First one's microfilaments. It is the thinnest. Maybe, if anything, know that. Right? Which of the 3 is the thinnest? Microfilaments. Which would almost be easy because it has it has micro in the word, but then again, so is the largest. So just think about, like, even though you're not biologists, think about what sounds bigger to you, a filament or a tube, right? So because filament, in my opinion, sounds smaller than a tube, and it has the word micro in it, then we can imagine it is the smallest of them. This is the thing that moves the cellular components. It also maintains structure of the microvilli, which we'll talk about later, maybe, because again, in the grand scheme of this semester, it's not very important. And it is common in muscle cells, and it is responsible for muscle cell contraction. So again, if anything, just know that of the three, this is the thinnest. Because again, this whole thing that we're talking about right now is not very important for the scheme of this semester. Obviously for life, it is, every single piece is important for life. But as far as what you need to know later in the semester, this is not very important. But I am kind of big on superlatives, right? So if I'm telling you something's the biggest or the smallest or the heaviest or whatever ist, then probably you might need to know it. So any questions about microfilaments? All right, the next one is, or are, intermediate filaments. These are intermediate diameter. So again, they're not the smallest like the one we just talked about. They're not the largest. These are the middle. Hence the name intermediate filaments. They have a structural function that helps maintain the shape cell. It helps anchor organelles, and some of them are made up of a protein called keratin. Again, none of that stuff would be helpful for us. So if anything, just know it's the intermediate. And once again, like I said, if anything, I'm going to be looking at superlatives, right? So the microfilaments are the smallest, as you might imagine once we get here. Microtubules are the biggest. So even though I might not ask you the middle, by process of elimination and by the name, intermediate, you should be able to remember it's the middle. The middle sized. Any questions about that? All right, like I said, microtubules are the thickest, and that is something that you should know. Well, if anything, that is the thing you should know for the exam. They're the thickest. Technically, they are hollow tubes. The rest of them are not hollow tubes. They are interesting, in my opinion, because they rapidly dissolve and reform. They also guide organelle movement, and they move chromosomes. So that part will be important later. We'll have a whole chapter about how chromosomes move later in the semester. And I'm only circling it for that reason. For this exam, I'm not going to ask you that, but later you will definitely need to know about microtubules, because without microtubules, the chromosomes don't move, and without the chromosomes moving, we can't have mitosis or meiosis, which is the whole thing that we discuss later. And then finally, those things I was telling you about earlier that help cells move, flagella and cilia, those are microtubules. Anyway, any questions about that slide? All right, here's a good comparing and contrasting. I'm not going to go through this, but if you download the PowerPoint, you can look at it, like to compel, to look at the difference between flagella and cilia. As far as I'm concerned, in this class, this 100 level non-biology majors class, you don't really need to know the difference. They're both just there to help them move. And if anything, the way I always remember it, flagella is the bigger one. There's like one or two big ones. And then the cilia, there's like a bunch of little tiny ones. That's the best way I remember it. And again, you can study that if you want, but I'm not going to ask you any questions about this on the exam. Actually, I was going to do some finishing touches on the exam, but one of your classmates has already taken it. So the exam is the exam. So I'll ask the expert. Is this stuff on the exam? Okay, just confirm. She knows more about the exam than I do at this point, because I wrote it a while ago, but I haven't reviewed it completely like I planned to. Anyway, moving past flagella and cilia, let's talk about the endomembrane system. And again, as far as I'm concerned, as far as this semester is concerned, this is not very important. We can go through it quickly because you could completely forget everything I'm about to tell you and understand the stuff I'm going to teach you later in the semester. That's why, in my opinion, it's not that important. But it is in the textbook, so I am talking about it very quickly. So it consists of membranes and organelles that work together to modify, package, and transport lipids and proteins. So in other words, our body makes lipids, our body makes proteins, and we're going to have a whole chapter on how our body makes proteins. That'll come later. And all we're saying is it's the endomembrane system that helps do these things, right? To modify them, to package them, to transport them. Obviously, something else makes them. It doesn't say the endo system makes them, right? Modifies them, packages, transports. And the pieces that the parts that make up the endomembrane system, right? It's the whole system. You have the nuclear envelope, you've got lysosomes, vesicles, the ER, the endoplasmic reticulum, and the Golgi apparatus. And this is interesting that you booked it this way, because so far, I haven't told you what any of these words mean, right? So we haven't learned those parts, but it's like, hey, here's the parts you've never heard of. That's the endomembrane system. So I guess you could look at it as a preview. So it's like, yeah, these are the parts of the endomembrane system, and I'm about to teach you about them. And again, as far as the exam is concerned, not important, right? So none of this was on the exam, right, Marks? All the cell parts, right? It's not the cell parts. Mostly chemistry, properties of life, definition of biology. This was really chapter 1 and chapter 2 stuff. Anyway, the nucleus. We'll talk about a lot in future chapters. So this is your first introduction. Nucleus is the most prominent organelle. So if we were to look at some slides of those microscopes back there of some cells, those would be pretty, you'd be like, oh, that's the nucleus, right? And these microscopes, I'm not talking about going 100x. I'm talking about, you don't even know what that means yet. I'm not talking about zooming in really far. Even just in the zoomed out look, you can see the nucleus. This is what houses the cell's DNA. You definitely need to know that later in the semester. That will be important. Not for the exam though, because that is so, it is important, but it's so easy to remember by the time we, later in the semester when we're done talking about it, that would be like too easy of a question for that chapter, because it's just that easy. Like, hey, where do you find the DNA? It's in the nucleus. And as you're going to learn in that chapter, what does the DNA do? Right? It directs the synthesis of ribosomes and proteins. Now that you already do know. You already know from the biological molecules section of chapter 2 that DNA, right? That's where we have genes, and genes are instructions on how to build proteins. So DNA is what's found in the nucleus, and obviously the nucleus directs the synthesis of proteins. And then, yes, this part's a little bit new for you. It also directs the synthesis of ribosomes. And not that you need to know that for this exam, but later you're going to need to know that ribosomes are actually the things that make the proteins. So the nucleus builds the machines, if you will, that makes the proteins. And then obviously that also helps the direction of the proteins themselves. Yep. Anyway, any questions about that? All right, and you know what? I wish I made this a little bit better. Not that this matters for this exam, but in a sense, this bullet point should be shifted over that way. Because this bullet point is describing what the DNA does, right? So the DNA is in the nucleus, and the DNA directs the synthesis of ribosomes and proteins. The nucleus is just where the DNA is. Does that make sense? Sort of like saying, the Pentagon is where we have, the military people that direct what the military does, right? But that doesn't necessarily mean those things, like the wars that are happening in the Pentagon, right? They're happening in wherever the war zone might be. Same here. The nucleus, it directs the synthesis of the ribosomes and proteins, but it's not necessarily happening in the nucleus. Any questions?
Synthesis just mean what they do?
Yeah. The DNA, the nucleus, actually, basically what I'm saying here, what I'm trying to say is the nucleus itself doesn't do anything, and it holds these things, it holds the DNA, and that's what the DNA does. It directs the synthesis of ribosomes and proteins. And as you're going to learn later, even that doesn't even happen inside the nucleus. The DNA is in the nucleus. The DNA that is making these calls is in the nucleus, but that doesn't happen in the nucleus. Just like the generals in the Pentagon might be saying, all right, drop a bomb on that country, but they're not doing it, right? They're in the Pentagon. It's not happening in the Pentagon. It's happening wherever they're directing it at. But again, for this exam, everything I just told you is details you don't need to know. And then for the next the exam, when this does matter, it'll be a lot less complicated. So we won't even talk. We'll just say this happens in the nucleus. This happens outside the nucleus. It'll be that simple. And for now, I don't even want to get into it because I don't want to muddy the waters. All right. The next word for attendance. Again, this is for a staff member that we lost. So the next word for attendance is Wendy, like the name, W-E-N-D-Y. Bless you. All right, so what is the nucleus made up of? Or what? How do you define what What defines it, maybe, I guess you could say? The nuclear envelope. It's a double membrane, right? So, the cell itself, as we know, has a membrane, which is one phospholipid membrane. But this thing has a double membrane, which is important for its function. I'll put it next to it here, though, but unimportant for this grand scheme of the semester. You can forget that it's a double membrane and still understand the things I'm going to teach you later. It is the outermost portion of the nucleus, obviously, and it has pores that control the passage of things, ions, molecules, RNA, and a big old X through this. So if anything, and it's kind of in the name, the only thing you would need to know, and like she's already confirmed, not on this exam, but the nuclear envelope is the outermost portion of the nuclear, which makes sense, but the nucleus, right? You've got the nucleus and then the membrane that's outside of it. That's the nuclear envelope. So still talking about the nucleus. What's inside the nucleus? If you're taking notes, you can ignore this next slide completely because we're going to talk about this in great detail later. Chromatin. We have a whole chapter based on chromatin. And we've already kind of talked about it a little bit in chapter 2 when we talked about the biological molecules and we talked about nucleic acids, right? And one of those nucleic acids we talked about was called chromatin, or DNA, right? So DNA makes up chromatin. Also, so does there are some proteins that help make up chromatin. I already talked about proteins in the previous chapter. But again, for this exam, you don't need to know this. This is just an introduction to something you're going to need to know later in the semester. So chromatin obviously makes up chromosomes. I shouldn't say obviously. Chromatin is the stuff that makes up chromosomes. What are chromosomes? Chromosomes are the hereditary material. They are linear in eukaryotes, meaning what I mean by that is like, I'm sure you guys have seen pictures of chromosomes. They kind of look like this, right? They kind of look like X's. That's what our chromosomes look like. That's what our duplicated chromosomes look like. But for a prokaryote, for example, it would actually be circular. And so, not that I'm going to ask you that, but yeah, there is a difference there. And again, you'll need to know this later. The chromatin, right? The chromosomes of a species have a specific number. That's part of one small part of how we identify species. Very, very small. There's a lot because some species do have the same chromosome number, but generally speaking, species have different numbers. Does anybody know how many chromosomes humans have by any chance? Not that you need to know yet, but you will later. We have 23 pairs. It might be a good independent work topic later, and I'll bring it up later too. Like, just think of some species and ask that question of it. How many chromosomes does a sea turtle have? Of course, there's different species of sea turtles, but how many chromosomes does a cat have? Anyway, any questions about this relatively unimportant slide as far as the exam is concerned? All right. Once again, relatively unimportant as far as the exam and the semester is concerned for this one. The nucleolus. That is the dark area inside the nucleus, and this is where ribosomes are made. And later in the semester, you will need to know about ribosomes, what they're made of, how they work, what they make. But one of the things we don't talk about, oddly enough, later in the semester are where they're made. So again, as far as the exam is concerned, as far as the semester is concerned, this is not important information. I might change this up next semester. I might throw some of these questions in the exam. How does this affect you? But just in case somebody's watching these videos in the future and they hear me say this, I don't want them thinking, oh, that's not going to be on the exam. My exams change from semester to semester, so never think because I say something on a video that it pertains to your semester. Any questions about that slide? All right, next, we're still talking about the same kind of stuff here. We're still talking about the endomembrane system. And the next part of the endomembrane system is the endoplasmic reticulum, shortened as the ER. These are interconnected membranous sacs, you might almost want to call them. That's all these things on the outside here. They modify proteins, and they do that in the rough ER because the rough ER has those little red dots, which are ribosomes. And the smooth ER, which doesn't have ribosomes, because ribosomes create proteins, they synthesize lipids. So again, we've already confirmed it from one of your classmates that none of these questions are gonna be on the exam. But if I were to put anything on the exam, I would say, all right, at the very least, you need to know that the ER is where we make, or modify proteins and make lipids. I would ask that. And then I might get so deep as to say, which one modifies proteins, rough ER or smooth ER? Which one synthesizes lipids, rough ER or smooth ER? But again, for this exam, not important. And again, we're gonna have a whole chapter on how proteins are made, how they're modified, all these things. All right, so any questions about this? All right, so now imagine what we've basically been talking about is a factory, right, to back it up a little bit. I've told you that the chromosomes are here, right? We've got the DNA inside the nucleus. The DNA provides instructions on how to build proteins. So then we build proteins, right? And then those proteins are modified here in the rough ER. Then they're ready to go, right? It's like, all right, we've got our proteins. They're modified. Now they need to go to wherever in the cell it is they need to be. And that's where we have this, the Golgi apparatus, AKA the Golgi body. It is a series of flattened membranous sacs. And these things sort, package, and tag proteins and lipids, right? So all the things that were made in the ER then go to the Golgi apparatus where they're basically ready to move, right? Obviously, the receiving side would face the ER because as it's leaving the ER, it's going to go into the Golgi body. And then the opposite of that, right, would go away from the nucleus. But once again, this is as far as the exam is concerned, as far as the rest of the semester is concerned, this is all unimportant information. Any questions about that? All right, lysosomes. They're basically known as the garbage disposal of the cells. So these things use digestive enzymes to basically break up the stuff that we talked about in the last chapter, right? So when you eat protein, very rarely, I would assume, I don't know this for a fact, I would have to look this up. Maybe I did know at one point. I would assume most of the proteins you need, your body doesn't necessarily need that specific protein. Probably what it needs is the amino acids that make that protein, right? So that's what these things do. They'll break it down. They'll break down that polypeptide into its amino acids. That way that your body can use those amino acids. Also, it tastes kind of like cleaning house. So our cells have organelles in them. When they're worn out, well, the lysosomes can break them down, right? Break them down to the pieces that build them up, and then use those pieces to build new stuff. And then finally, they destroy disease-causing organisms. So, for example, if you get a parasite that gets in your cell, like that's how the malaria parasite works. In theory, a lysosome should destroy that. Viruses, which aren't alive, so they're not organisms necessarily, but in theory, when a virus inserts its genetic material into your cell, the lysosomes should destroy them. Obviously, that doesn't always happen, otherwise people would get sick from parasites and bacteria and viruses. But once again, as far as the exam is concerned, as far as the semester is concerned, this is unimportant information. Any questions about this slide? All right, the next one on the list, vesicles and vacuoles. Once again, not important as far as the exam or the semester is concerned, so let's go through it quickly. These are membrane-bound sacs, which is another way of saying, the way I like to think of it is like you've got the cell that has a big old membrane, right? Then inside of it, you have smaller little, I want to say cells, because it is, when you're looking at what a cell looks like in that context, right? Yeah, these are like smaller cells. They are in that they have a membrane and they're like smaller little versions of the cells, except they don't have all the stuff the cells have inside of them, right? They don't have a cytoskeleton and organelles. But they're just like, as far as the membrane is concerned, they're just like a smaller little version of the cell. They are used for storage and transport. Sometimes there's vacuoles that are larger, vacuoles are larger than vesicles, and because of that, they can break down macromolecules as well. Right? And sometimes vesicles fuse with membranes. That's how we have things leaving the cell, actually even sometimes coming into the cell. But again, as far as the exam and the semester is concerned, this is relatively unimportant information. Any questions about this slide? All right. Next are the ribosomes. Very important for the semester, not important for the exam, because again, there'll be a portion of the semester where we talk about this in great detail. Ribosomes are the things that make proteins. They're made up of a large and small subunit. We need to know that later, but not for this chapter, or excuse me, this exam. And again, they're going to be discussed in later chapters. So for now, we'll just leave it at that. But yeah, we're going to learn a lot about ribosomes. And obviously, they're so important that we've already heard about them, right? The thing that defines the rough ER is the fact that there's ribosomes on it. As opposed to the smooth ER, which doesn't have ribosomes, right? So rough ER is where the proteins are modified, right? Because ribosomes make proteins. And the smooth ER doesn't have ribosomes, and that's where lipids are synthesized. Any questions about ribosomes? All right, the next one, we're going to have a whole chapter on this one too, so we're going to go through it pretty quickly once again. As far as this exam is concerned, this is very unimportant. As far as the semester is concerned, this is very important because we will talk about it later. A lot of people call it the powerhouse of the cell. So all the energy that your cell needs, not all of it, but a huge majority of the energy that your cell needs comes from the mitochondria. Even plant cells, like let me ask you this, forget that I'm talking about this for a second. If a kid were to ask you, where do plant cells get their energy, what would your answer be? That would be good too, because that is, like I told you earlier, starch is how plants store energy. But yeah, then you are correct in that. But again, think about it like just a kid asking you, because then you'd have to explain where starch comes from and all that. If a kid's asking you, where do plant cells get their energy? Plant cells, not animal cells, but plant cells. A leaf, okay. And that's a leaf has plant cells. So where do those cells inside the leaf get their energy? The sun? Yes, that's what I'm getting at, right? Most kids think, oh, the plants get energy from the sun. And they do. But even then, the cells inside of plant cells, it's not like they just get the sun energy and they just function without all the other stuff. They do the same thing we do. They have the mitochondria that takes down carbohydrates and things like that and breaks them down to release their energy. So even plant cells don't even get sun energy directly from the sun. They use that energy to create glucose, amongst other things. And then that glucose goes into their mitochondria where it's broken down to release energy. So even for plant cells, this is where they get their energy. I also want to point out, this is a great time to point this out. Not a great time. I should have been pointing out this whole time, reminding you. We are talking about eukaryotic cells, right? Not prokaryotic cells. And remember, one of the big differences between the two is the eukaryotic cells have organelles and prokaryotic cells don't. And this is what we've been talking about, it's organelles. So it makes sense that we're talking about eukaryotic cells. Anyway, again, really quickly, we're going to talk about this later in the semester. So for this exam, this is unimportant. For the whole semester, it is important. Mitochondria is the powerhouse. Why do we call it that? Because like I said earlier, this is where we get our energy. And specifically, the type of energy that we use is ATP. Right? So, there's just different forms of energy. Like, hey, what kind of energy does this computer work with? Electricity. What kind of energy does my old pickup truck use? Gasoline energy, right? So there's different types of energy. Well, the cell's energy is ATP. Mitochondria are also double membrane. In this case, that is not trivial. That will be important later in the semester, and we'll talk about why that's important later in the semester. Another thing that makes them very interesting is they have their own DNA. Interesting, but not important for the exam or the semester. But they have their own DNA. So have any of you done like a 23andMe or anything like that? Well, anyway, it's interesting because you had your dad's sperm cell met with your mom's egg cell. Sperm cells don't have mitochondria. So your mitochondrial DNA came only from your mom, which came only from her mom, which came only from her mom. So it's really interesting where they can trace your heritage back when they're looking at the mitochondrial DNA, because it's not mixed with any other stuff. It's your mom's, right, all the way back, which I think is, to me, it's just fascinating stuff. But again, as far as the exam and the semester is concerned, not very important. All right, so there's no questions about mitochondria. Let's talk about peroxisomes. These are organelles that only have a single membrane as opposed to double membrane, like the nucleus and the mitochondria. And there's another one that we're talking about later that also has a double membrane. But these things are the things that break down fatty acids and amino acids. And they also detoxify many poisons. Once again, for the exam, for this semester, this is another unimportant one because we're not going to bring it back up. You can brain dump it when you leave this room, and you should be okay for the exam and for the rest of the semester. But again, I teach out of the textbook, so I didn't want to just not talk about it because then when you're studying your textbook, and I'm sure you guys often do, you can read this and be like, I remember him saying, as far as the exam and semester is concerned, this isn't important. All right, so that's it for the parts. Let's do a little comparing and contrasting some animal cells versus plant cells. And mind you, there's other stuff too, right? We're talking about eukaryotes here. There's plants, there's animals, there's fungi, there's things like that, right? So everything I'm telling you right now is comparing animal cells to plant cells. So if you want to, for independent work, you can look up, well, what about fungi, for example, right? So anyway, plant cells, they have no centrioles, they have no centrosomes, they have no lysosomes. Animal cells do have those things. Plant cells have cell walls, they have chloroplasts, they have something called plasmodesmata, which I'm going to teach you about later, but even then, once I teach you about it, it still won't be that important. Same thing with plastids, and they also have a central vacuole. Animal cells don't have those things. And again, as has been confirmed, these questions will not be on the exam. But in a different semester, I might have somebody recognize the difference between plant and animal cells. And just like I mentioned before, it's a 100 level class, multiple choice. So instead of saying, tell me the difference between plant and animal cells, it would be more like, I would say, this particular cell has no centrioles, but it does have a cell wall. Is that a plant cell or an animal cell, right? So you would just need to recognize one or the other based on the description. So that being said, let's dive a little bit more into plant cells. Like I said, plant cells have cell walls, animal cells don't. So let's talk about that very quickly, because again, for the exam and for the rest of the semester, this won't be as important. We will bring up cell walls again later when we talk about cellular division briefly. We'll talk about it then. I think that's about it. Anyway, cell walls are external to the plasma membrane. So again, think of just a cell. Well, you have that cell, the plant cell, is now encased in the cell wall, unlike the animal cell. It protects the cell, obviously. It provides it structural support, obviously. It gets its shape, obviously. I think these things are all very obvious, right? It's a wall. Walls do these things, right? Even if we're talking about non-biology stuff, talking about a house. Well, the wall protects the house. The wall gives the house structural support. The walls give the house shape, right? All these things that you think of as a wall. Same thing here. Of course, where that, I was going to say it's a little bit different, but this is kind of true too. So the cell wall is mostly cellulose. Hopefully you remember from chapter 2, and you're going to need to know from chapter 2 for the exam. Cellulose is a carbohydrate, right? It's a bunch of glucose monomers put together, and that is what the cell wall is made up of. And the reason I kind of chuckled is, now that I think about it, a lot of houses, you've got 2x4s inside the wall. 2x4 is made of wood. That's almost entirely cellulose. So yes, even your walls, depending on how you look at it, your house walls are also mostly cellulose, depending on how you look at it. Anyway, fungi and protists also have cell walls, but they're not cellulose. So you can look that up for independent work if you want. And again, it's a good time to remind you, if you're just answering a question with like a one-sentence answer, you don't need to write a paper. You can just send me an email, not as an attachment, just an email that says, I looked it up, and according to this source, fungi cell walls are made up of, right? If you're just writing a one-sentence answer just to get 1 point, just email it to me. Now, again, if you were to look it up, I looked it up, and fungi cell walls are made of this, and the reason, if you start writing about it, then you might want to write me a paper. Also, another thing I would recommend is, even when I give you guys these ideas, wait until after class before you do these. Because obviously, if you're going off on a tangent, something that's relatively unimportant, then you're going to miss something important, I might say. Anyway, any questions about cell walls? All right, so speaking of the wall, the next phrase for attendance is Pink Floyd. Pink Floyd. Extra credit? No. Okay, Okay, chloroplast is the next part. So again, these are parts that are in plant cells but not animal cells. Chloroplast. This is where photosynthesis happens, which is why for this exam this is important, because later in the semester we're going to talk a lot about chloroplast. This is the other double membrane organelle, along with the mitochondria and the nucleus, and that again will be discussed in later chapters. So for now, for this exam, unimportant. The double membrane thing again, that actually is important. It's not trivial. So the double membrane, the fact that chloroplasts have double membranes, the fact that mitochondria have double membranes, that's actually important because they won't be able to do what they do without the double membrane. So that will be important for the grand scheme of the semester. What's not important for the grand scheme of the semester, if you were to look it up for independent work, is the theory, I think it's a theory now, not a hypothesis, the theory of why these things have double membranes. So there's a reason why mitochondria have double membranes. And I don't mean like, how do I explain this? I don't want to confuse you. I just said that they're double membrane and that has a function, it's an important function, but I'm saying the origin, like where did that come from? Why? How'd that happen? So there's some interesting reasons why chloroplasts and mitochondria have a double membrane. Not why they're used, but how it happened. And you can look that up in independent work. Any questions about chloroplasts? Central vacuole is the next thing we're going to talk about. Again, this is only in plant cells. This actually takes up most of the cell. So when you're looking at a plant cell, most of it is the central vacuole. This is the thing that regulates the water concentration. So later in this chapter or the next one, I forget because it's kind of shuffled, slightly different organization from the last textbook that I've been using for years. Anyway, later we're going to talk about water concentration. As you should know if you've done your pre-lab, which is due, by the way, today by midnight. You're going to learn about a little bit about water. Let me see. Yeah, diffusion osmosis, right? So this, at least for a plant cell, is very important for stuff like that. Oh, which brings me up to a point. Thank goodness. One of your classmates did something very smart and came to me for help, which I recommend you doing. By the way, I have office hours today until noon. So when you're doing your pre-lab, come to me for help. But I want to point some things out so you don't make any mistakes. A hypothesis is just simply an answer to a question. Technically, like what we said, it's a falsifiable, testable answer to a question. But when you're answering a hypothesis question on the pre-lab, just remember it's just an answer to a question. So if I say, using the last one as an example, biological molecules, what do you think the monosaccharide content of starch is? For that, it's just simply a little bit, none, or a lot, right? It's nice and simple. So for this pre-lab, I might say something, hey, do you think water is going to enter the cell or not? Yes or no? Do you think iodine is going to enter the cell or not? Yes or no? Do you think starch is going to leave the cell? Yes or no, right? Don't overthink it, keep it simple. Now, if you do the predictions for extra credit, remember the prediction, that's when you use the if-then statements. And that's when you describe basically how you're going to test your hypothesis. So again, just using this one example, I know I'm going off on a tangent, but you guys haven't pre-lab today, so I want to make sure you understand here. If you hypothesize, in that one experiment when you put the little fake cell that has starch solution in a beaker of pure water, if you hypothesize, if your hypothesis is that starch will leave the cell, then your prediction would be if starch leaves the cell, then the water in the beaker will turn dark purple, right? Because you put iodine in there. Like, so you're describing how you would know it, right? So your hypothesis is just saying, I think this will happen. Your prediction describes how you're going to know it, right? Anyway, and if that confuses you, just don't do the extra credit stuff. Leave the predictions out of it and stick with the hypotheses. Or take a shot at it because it's extra credit. So if you screw up the predictions, you're not going to lose any points. Just might not get any or get very few. Anyway, yeah, so that's the central vacuole. We'll talk about it later. Actually, a little bit later. And we won't even talk about it that much to begin with when that happens. Any questions about the central vacuole? Okay, another very unimportant one as far as the semester and the exams are concerned is something called an extracellular matrix. As you might imagine by the name, it's something on the outside of the cell, right? And it's mostly something called glycoproteins and collagen. that are then actually secreted from the cell. And this is the thing that basically holds cells together. And as you might remember, even though I'm not going to test you on it, but as you might remember from chapter 1, when we talked about how it goes from atom to molecule to organelle to cell to tissue, right? This is what basically makes tissue happy, because this is what holds our cells together. Which is actually, so again, think about life. As far as life is concerned, this is very important. So even though we're not going to get tested on it, and even though you could brain dump it and be fine for the rest of the semester, we wouldn't exist without this, right? We would just be a puddle of cells that are not connected to each other. And also, because they're connected, that's also how they communicate.
That's the animal cell wall.
Great question. It's not necessarily the cell wall. It's in the membrane. I just hesitate to call it a wall, but it is so it's in the membrane of the animal cell. Good question. Any other questions? Excuse me. On a semi-related topic, and once again, unimportant for the exam, unimportant for the rest of the semester, we have something called intercellular junctions. So intercellular means in between cells, and junction is just what it sounds like, right? Where things come together. Intercellular junctions are direct contact between cells for communication. We have something called plasmamesdata junctions that are between plant cells. And I mentioned that earlier, and I said you didn't need to know it. And once again, you still don't need to know it. For animal cells, the contacts include tight and gap junctions and desmosomes. You don't need to know any of this. Here's a picture of what I'm talking about. This is from your textbook. So you can see these plant cells right here. Those cells are connected, but there is this little plasmodesmata where things can kind of go back and forth. You can see the groups of tight junction proteins in these animal cells. All this stuff for us is not important. Once again, you can brain dump it and still learn everything that I'm going to teach you later in the semester. So for those reasons, not important. Now, if you were going to be a biologist, you would need to know this stuff. So another thing too, speaking of which, if you were going to look up. If you were going to look up like how a virus works, a lot of times a virus is attached to a cell, and they only attach in certain spots. So in that context, this stuff will be information, relatively important information. But once again, you've been exposed to it. So now it's up to you later in your life. If for some reason you did want to learn more about it, go for it. The good news is your textbook's free, right? So even though you leave the class, you still always have access to that textbook. And it updates, right? So that's a thing that updates. As things change in biology and we make new discoveries, that textbook will be updated. All right, so that's it for eukaryotic cells. Now we'll talk about the cell membrane, or at least start that conversation. Hopefully get you all excited. And then over the weekend, you'll be like, oh man, I can't wait to come back on Monday and learn about cell membranes. Forget this weekend, it's so boring. Anyway, learning objectives for this section. At the end, you should be able to understand the fluid mosaic model of membranes, which you will not need to know for the rest of the semester. And you should be able to describe the functions of phospholipids, proteins, and carbohydrates in membranes, which you will not need to know for the rest of the semester. Let's keep going. I'm trying to get us caught up here. The fluid mosaic model, like I said, you need to know, technically this is the learning objective. You need to know what this is. It describes the structure of the plasma membrane as a mosaic of fluid components. How many of you outside of biology know what a mosaic is? Like, if we were in an art class, what's a mosaic? Yes?
It's like a bunch of random patterns.
Yeah, a bunch of random patterns. Sort of like, I guess you could, if you wanted to, maybe get a bunch of broken plates, maybe, and like plaster them together and make make something pretty out of it. Random stuff put together, right, for a larger function. That's what this is. So the mosaic model, I've already told you that we have this membrane, this phospholipid membrane on the outside of our cell. Now I'm just getting a little bit deeper and I'm telling you, it's not just phospholipid bilayer, there's also stuff inside of it, hence the mosaic part. And it's fluid, meaning for the most part, they're not in one spot, they kind of just move around within the membrane. So fluid, mosaic, that's what that means. But again, as far as the exam is concerned, as far as the semester is concerned, that's not very important. But again, this goes back to one of the things I was saying earlier. You see it's on glycoproteins, glycolipids. Remember I was telling you viruses very often will attach to a cell. And sometimes that's the way they do it. I don't remember the specifics, but the virus that causes COVID-19, it has a specific thing that it, attaches to. Anyway, so again, that could be important later in your life, and this is why we're just giving you the base information for you to look up later if you want. Fluid mosaic model. All right, animal cells have cholesterol in our membrane, which is why, when you eat animals, it's usually higher in cholesterol. What does the cholesterol do? This would be a good test question if it was on the exam, but it's not. The cholesterol regulates the fluidity. It kind of serves as an antifreeze. Now, don't get me wrong, that doesn't mean that cells can't freeze, but that just helps them freeze slower or freeze at a lower temperature, right? Yeah, but that's basically it as far as we're concerned. It has other functions, but those are the big functions. Any questions about that? All right, again, like I've already said before, the membrane is mostly a phospholipid bilayer. As I said before, and I think you asked about this, the outside is hydrophilic, the inside is hydrophobic, which is important for how things function. And if you've done the pre-lab, I'm pretty sure this information was included in that video. I don't remember for sure, but anyway, it is important for functioning, but as far as the exam is concerned, I'm not going to ask you that. Any questions about that slide? Okay. So again, if we're talking about the cell membrane, the largest component is the phospholipid bilayer. The second largest component are proteins. And the proteins have a lot of functions. Sometimes they can be channels or pumps, which you definitely learned about in the pre-lab video, because when we talk about diffusion, things can come in and out of cells simply. But some things are too big or charged, and they need a pump or a channel to get through the membrane. They serve as enzymes that are sometimes attached or embedded into the membrane, sometimes not. But in this case, we're talking about the ones that are embedded into the membrane. They also serve as attachments for the cytoskeleton. And finally, cell-to-cell recognition, right? So it's how some cells can determine, this cell should be in my body, or this cell should not be in my body. The third major component are carbohydrates. These are always found on the outside of the cell membrane. They're always bound to lipids or proteins, and once again, they're used for cell recognition. That's perfect. None of that will be on the exam. None of that's going to be important later in the semester, so you can completely brain dump the thing we just finished, which is 3.4, and then when we come back on Monday, we will talk about the last word for attendance, which is passive. That's the last word for attendance, but what we're going to talk about is passive transport on Monday. You guys have a good weekend, and hopefully you'll visit me online for office hours, and I can help you with your assignment before you turn it in.